All right, hello everyone. I'm Robert Petit. I'm from the Wyoming Public Health Laboratory. It's out west here in the US, one of those rectangle states. But yeah, I'll be talking about some enhancements I made to Bactopia for ongoing genomic surveillance. So let's start off. What is Bactopia? Well, it's an end-to-end Nextflow pipeline for the complete analysis of bacterial genomes. In just a few simple steps, you can go from your raw sequencing data to just a crap ton of results to sift through. Literally sometimes. Sequencing, so it started at step one. You can sequence your bacterial genomes. It accepts Illumina and nanopore data, or you can use a combination of the two. And then at step two, you got to install Bactopia. It's easily installed through Conda, and your analyses can be done through Conda, Docker, or Singularity. And at step three, there's a prepare command to create a sample sheet if you need to that allows you to process a single or tens of thousands of genomes, whatever you need. And then step four, the previous lab I worked with, we utilize public data a lot. So there's a search command that allows you to query ENA and SRA and automatically find matching accessions and download those and process them in Bactopia. And then, it's built using Nextflow, so all the executors that Nextflow supports, and it supports a lot, Bactopia supports those as well. So with simple parameter changes, you can go from playing on your laptop to your HPC to any of the major cloud platforms. And then step six, we're finally processing your genomes. Bactopia includes more than 150 bioinformatic tools into the pipeline. Its goal is to be end-to-end. There's extensive version control and data provenance. And the idea is after this initial run, you have all your outputs in a nice standardized directory structure that you can then pass to the Bactopia tools. And the Bactopia tools are kind of my favorite part. These are 60 additional workflows that you can make use of the already existing outputs. These allow you to do complex analyses relatively quick and with little effort. That includes things like pangenome analysis in bacteria, which is a bit different than the human pangenome, which we'll learn about here shortly. But you can also do taxonomic classifications, create phylogenies, do all your AMR and all sorts of species-specific analyses directly from those outputs with little effort. And then finally at step 8, you're stuck investigating all the results so you can write that paper up at the end. Or let your epidemiologist know what's going on. But so just to kind of sum it up, Bactopia streamlines the analysis, enabling users to focus on their science and outcomes, which is very important for me because most of the users I work with of Bactopia are not bioinformaticians. They're just people who've been getting data that they need to process. They're already up to their necks and just sinking. And so this idea of making it easy and keep moving is essential for me. But as the bioinformatician, I recognize the need for some revamps in Bactopia. This is the main pipeline in version 2 of Bactopia. It includes all the essential bacterial data analysis steps, your QC and reads, assembling genomes, annotation, sequence typing. But it only supported one species per run. You could run any bacterial species on it, but you couldn't run them together. So you couldn't process your salmonellas with your E. coli, so you had to run them separately, which is inconvenient when you're getting multiple different species coming into your lab to be sequenced for ongoing surveillance. It was also very difficult to merge the results from multiple runs, because the idea was you would already have all your data, and then you would analyze it and do your work. Ongoing surveillance, you're getting data over time, and we can't wait to notify our epidemiologists that, hey, there's an outbreak going on, but I'm waiting, a year from now to acquire all the data to analyze it. There was also a number of steps that relied on input data sets. There was a script to automate this. But yeah, that's a lot to ask for new users. Hey, you need to build all these data sets before you can run Bactopia with all these additional things. Also, if you're doing multiple species, you need to create a data set for each species that you're working with. I also maintain the Docker containers and Conda environments for each one of these steps, and that can get quite tedious over time. And not all modules were true plug and play. All the Bactopia tools, they're available from NF Core modules, so they follow that NF Core modules practice. These modules did not. These were kind of the core ones that didn't quite get converted when I released version 2. So to give you an idea, a simple genome here, it expected the FASTQ files to come from this QC read step. And you couldn't really move these around. And that is way too strict for me. With all this, it made the version updates just a huge hassle. If I needed to make a quick patch, sometimes it's easier just to ask the user to go, hey, change this line of code, which is not the way to do it. But if it could save me a weekend, please just change this line of code. So I put myself in this situation where this flame was just growing, and it was not fine. So I started working on Bactopia version 3 with some main goals that I wanted to meet Wyoming's public health pathogen surveillance needs, while also lowering the barrier of entry for new users, and reduce the long-term maintenance burden on myself. That's probably the biggest thing I was aiming for here. So by doing this, I streamlined the pipeline. We still include the main steps, QC, assemble, sequence type, annotation. But what's nice now is you can mix and match any number of species per run. You can mix your salmonellas with your E. coli, with your streptococcus, with your staph aureus. There's no need to run separate runs for any of them anymore. It's also easy to use the results from multiple runs. So since we're getting samples over time, I changed it to where now each sample contains all the data it needs. All the results for each sample is self-contained, allowing you to move them around and compare across runs, no problem. The data sets are also baked into the process. Users no longer have to go and build data sets. This is automatically handled now, which is, again, a nice, they can just go straight to processing data instead of, oh, by the way, you need to run this first. I also delegated the containers and environments to Conda because the infrastructure is there, and why not take advantage of it? And all modules are now plug and play. So all modules now follow the NF Core modules best practices. So all these modules here that you see are treated just like the 60+ Bactopia tools. And overall, this makes the whole version update easy and seamless. If I need to make a patch, I make a patch. There's no sort of, oh, do I need to spend the whole weekend working on this, sorting this stuff out? So in my mind, at least, I had essentially flipped the scenario. I would much prefer to be in a room full of puppies than a burning room. So I truly believe I met that, but I really quick wanted to explore this plug and play bit for Bactopia. So all these modules are plug and play. There's 30+ not listed here. But for each module, I package a Nextflow config file and a params.json file. So it'll automatically pull in the corresponding config file and the params.json, which will feed into the CLI, which is taken from the NF Core template there. But in my mind, I can start treating all these as building blocks. For example, and this is a question I've been asked, I don't want to run the whole Bactopia pipeline. Can I just take pieces of it and do stuff with it? So for example, one I got recently was, I just want to call SNPs and indels against this reference for all these samples. I don't want to assemble the genome. I don't want to annotate all that fun stuff. So essentially, you could take the gather module, the QC module, and the SNP module. And now you can essentially skip all that extra stuff and just go straight to calling reference, calling SNPs and indels. Additionally, if you wanted to look at insertion elements, you could toss in the IS mapper module as well. And so now you can kind of start picturing how we can rebuild stuff and all that fun stuff. To give you an idea, I reshuffled some modules for some new named workflows in Bactopia. The first one is clean your reads. And this idea is I don't want to run all of Bactopia, I just want to QC some reads with the addition of removing host reads from there. So yeah, it's just a simple pipeline, gather your samples, scrub the reads, and then QC them. But the nice thing is you still get access to all those additional Bactopia tools without having run the full pipeline. So if you wanted to, we could replicate that snippy thing by just running the clean your reads and then the snippy Bactopia tool afterwards. In addition, I have one called Teton, which is named after the Grand Teton Ranges in Wyoming. But this is another simple one where we want to scrub the reads to make our Office of Privacy and Security happy, so we can confidently say, hey, we did the best we could to remove all human reads from these samples. And then do some basic taxonomic classifications. I underscored basic there because at this point, I just want to know, did I sequence what I thought I sequenced? And also, is there any potential contamination in here? And so we run this on all our newly sequenced samples to where they hit the cloud infrastructure, and then they get scrubbed, and we get a basic QC on taxon information before we run anything. So that would apply to our viral, fungal, and bacterial samples. So there should be some folks in the audience, how's this any different from NF Core tools and using NF Core tools to rebuild stuff with the modules? And so for me at least, this is a way to allow people to reframe Bactopia without having to relearn a new toolkit. In my field, at least in public health, we're usually understaffed. And having to retrain folks can be quite difficult. And by being able to give them a mechanism to reshuffle modules, they can now do all this without having to relearn a new toolkit. Because cleaner reads and Teton take the same inputs. If you know how to use Bactopia, you know how to use cleaner reads and Teton. And that's the idea behind here, where we can reduce that training time. So yeah, let's go ahead and wrap this up. Bactopia version 3 has been rebuilt for ongoing genomic surveillance of public health pathogens while improving the user experience and significantly reducing long-term maintenance. I think users tend to be happier with this new version. It seems easier to use. But me personally, I no longer feel like I'm in that burn-in room. I feel like there has been a significant maintenance burden reduced in Bactopia, which is quite nice. Briefly, a lot of people to thank, many of the folks in this room. All the developers of open source software, there's so many. On the doc site, you can go and get links to all of them. And the many users who are regularly providing feedback and suggestions. The Conda folks, Nextflow, NF Core, Sakara. My colleagues from Wyoming Public Health Laboratory, Taylor, Chase, Jim, Rob, and Joe. Taylor and Chase give me great usability feedback, which is super awesome to get. Tim Reed and the Emergent Group over at Emory University, and all these GitHub users who provided feedback while I was developing version 3 on, what could we change, what could we improve. And then finally, the Georgia Emergent Infections Program and the Centers for Applied Pathogen Epidemiology. Thank you. With that, I'll take any questions.
Great, thanks a lot, Robert. It's been fantastic to see public health in terms of their adoption of Nextflow, and it's really driving things forward. We see a lot in the CDC with some of the public health labs as well. I think the work that you're doing is really making a big impact there, and it's really changing folks as well. Got a couple of questions here. It's really good to see the convergence with NF Core related to the modules. Can you provide us with a kind of a quick summary of what are the key differences though between Bactopia v3 versus like a standard NF Core pipeline, and maybe some of the design decisions that you made for that?
Yeah, so I think I have a bit more flexibility because I'm not trying to fit a bunch of different pipelines into a single framework. I do add some stuff to the modules. That doesn't mean it's better or worse. It's just different, including additional login information and some additional version information. But I think it's one of those where if I were building this pipeline in 2023, I wouldn't have done it this way. I would have gone to NF Core tools and downloaded all the modules I'd need and glued them together. Unfortunately, this goes back a long time, and so I didn't have that option.
Okay, that's good to hear. And what do you see as the main challenges today, even with version 3 that you've developed, for biologists to use Bactopia or kind of similar pipelines in a more autonomous way?
Maybe this kind of initial fear of the command line. So each of the people I work with in Wyoming, they all are quite comfortable with the command line. And I think once you get over this initial fear of the command line, this black box, you open your world to a whole slew of different things. And there's obviously those, hey, I'm trying to work on this HPC system, but that's universal to everybody because each HPC system has their own restrictions, and that's just kind of, we just take it a comment at a time on the GitHub issues.
Cool. And final one, related. How do you see that what the work that pipeline developers can do to make those pipelines more accessible? And what's the kind of steps that you take in the development of Bactopia for making it more accessible?
Make it easy to use. The attention span is going to be short. If I have to go and build a bunch of things, it's difficult to install. That's going to put a huge hurdle on actually wanting to use it. For me at least, yeah, making it easy to install. Taking the NF Core module approach, because a requirement for NF Core modules is it has to be on BioConda, or it has to be available from Conda Forge. You've already solved the installation issue. And then that you have the containers and all that. But yeah, make it something that you yourself are going to use as well, so that way you find the bugs. And yeah, something that you feel like you can hand to other people and allow them to break it. I enjoy stuff getting broken. So that way we can fix it and figure out why.
Awesome. Well, good luck with the rest of your weekends then. Yeah. Awesome. Thanks a lot, Robert.
Thank you.